Hello, this is Mr. Buss, and I'm going to go through this transcription translation practice worksheet. We did this in class, of course, and I feel like it's a very important document, and it's going to teach you how to do both the processes of transcription and translation, so I wanted to make sure we had a video resource for it. I'm actually going to skip the initial pictures and move right down into here because we'll talk about those pictures in just a minute, but we're not ready to yet. All right, so first of all, transcription is the process where DNA is converted into the molecule called RNA, ribonucleic acid, okay? And that happens in the nucleus, right? Because DNA is trapped, it's a large macromolecule, double-stranded DNA, many thousands and even millions of nucleotides, A's, T's, G's, and C's long. And transcription is necessary, again, because, I'm not going to write it all out, but I'm just going to explain it, because DNA can't leave. But the code of life, right, the code, the ATGC code that is specific to specific genes needs to get out. And so the RNA is the trick. RNA is small enough to leave through the nuclear pores. Transcribe the DNA strand into mRNA. So here's kind of a typical question you'll see. Now, we know that DNA is double-stranded, right? So you might ask yourself, why is this single-stranded? Why is there not two strands? Well, because only one side of the DNA is the template strand for RNA. So the other side is not necessary, okay? And so also you might say, well, Mr. Buss, I know that if we're going to go from left to right, I know that DNA is made in replication in the 5-3 direction. Why is this backwards? Well, it's not backwards, because we're going to make the mRNA strand in this direction, and this is the 5' side because it's opposite. So we are still making 5-3 directionality. So below this thymine for DNA, I'm going to put A for adenine. Below the A for DNA, I'm going to put T. Just kidding, right? Remember that we can't use T's in RNA, so I'm going to put U. Apples under trees. And below C, we still have G. And below G, we still have C. So that's how you do your base pairing rules. So pause the video for a moment, make sure that you're able to do this. Go ahead and fill out the rest of this. I'm going to fill it out right now, though. Right? 3'. So that is our transcription of a DNA template strand into mRNA. Not everyone might remember what the M stands for there. That stands for messenger. Messenger RNA. We are going to have different RNAs that we're going to learn about, like transfer RNA and ribosomal RNA. Okay, so the next step, the RNA is going to leave the nucleus after it's transcribed. And then the RNA is going to then find a ribosome, or a ribosome is going to find it. Remember the ribosomes when we studied cells? Ribosomes are made of rRNA and proteins, and they are either found free-floating in the cytosol, or if you're a eukaryote, then it could be attached to the endoplasmic reticulum. And it's turning RNA into protein sequences of amino acids. Amino acid sequence. Okay. Translation happens again at the ribosome. I already said that. So you have to be careful. There's two words that start with a T that sound very similar. Transcribe and translate. Okay, transcribe is the first part. Translate is like a new language. We're going to go from a nucleic acid code to a amino acid code. So it's like a different language. That's why they use the word translate. So here's what we do. We're going to go ahead and break this code up into triplets or codons. Every 3. So every 3, oops, that wasn't very good. Every 3 of these is a codon. So there's 5 codons that I broke up the sequence into. 1, 2, 3, 4, 5. 5 codons that we're going to translate. So I take a look at the first one, AUG. okay? And I'm going to read this in the 5 to 3 direction, AUG. So on the left here of this grid, this is also on page 197. This is the first letter, okay? So I'm going to use this column, whoops, right here. And then on the top, it says second letter, so AU, and that means that I'm going to be in this region. And AUG is right here. Oh, look at that, it's the start codon, right? Which makes sense because I'm starting the amino acid. So the start codon AUG, and this is an mRNA code, right? That's important to remember that we are using this chart for mRNA. I look up the mRNA codon. So AUG codes for methionine, MET. I'm just going to write MET. I'm not going to write the whole word methionine. You could. I'm just going to write MET. Okay. If we wanted to use this, it's the same idea. Okay. I start in the middle though. So A, and that puts me in this quadrant. U, and that puts me in this quadrant. G, right? So that's how to do the code wheel, AUG methionine, okay? And again, the code wheel, that's only giving you the 3-letter abbreviation. Sometimes amino acids don't, the first 3 letters of the amino acid is not the 3-letter code, but that's okay. I'm not going to make you memorize all the ones that are slightly different, like, like tryptophan, TRY, is actually TRP. So there's a few that are slightly different, but we're not going to worry about that. So let's do the next one, CCU. CCU, proline. UUU, phenylalanine, PHE. UCU, UCU, serine, SER, and UAG, UAG, what does that mean? Stop, okay? So that means that's actually not an amino acid. Stop means stop. So that means it's done. Okay, that is how to do transcription and translation. So now you should be able to do most of this worksheet, to be honest with you. The only one last thing I want to talk about before I stop this video is we need to understand what tRNA is and how it works, because there are a few questions that use tRNA, okay? So when we have mRNA that's made, when it's transcribed in the nucleus and it leaves the nucleus, this is going to be part of a ribosome. There's 2 subunits, the small subunit and the large subunit. Okay, and it's going to attach pretty much close to the start codon, which is AUG. Remember, that's methionine. Okay, so when the small subunit attaches, something called a transfer RNA, a tRNA, comes in and matches up with the codon. Okay, so this is important to understand because methionine cannot recognize the start codon AUG. Methionine is an amino acid, so it doesn't have any affinity or connection with AUG. So we need this intermediate molecule called tRNA that has 2 functional ends. One end has an anti-codon, which just means complementary base pairs, right? A, U, G, and C are going to match up complementary. So A to U, U to A, and G to C, okay? And then tRNA also has attached to it the correct amino acid, methionine, right? And so the tRNA that has the UAC anticodon will have methionine attached to it. And so again, the purpose of the tRNA is to attach to the ribosome. So this whole thing now is the combined ribosome of the small and large subunit. It clamps onto the mRNA. And then these tRNAs are going to match up complementary anticodons to codons. And as the ribosome moves along, this is going to go to the next codon and the next codon and the next codon and so on. And the right tRNAs are going to bring the right amino acids, okay? So when you get to questions on this document, like this one that have you deal with tRNAs, so I'll do one of the columns here. I'll do this column. So if I have a DNA triplet GAT, complementary mRNA would be C to G, right? A goes to U, because it's RNA, and T goes to A, right? So C to G, A to U, T to A. That would be how to make an mRNA. But then what is the anticodon that would be complementary to CUA? Of course, it would be G, A, U. Notice that the DNA and the tRNA anticodon are almost identical, except, of course, I can't use T's, I have to use U's. But then if I want to know what the amino acid is, what do I look up? Well, remember, I look up the mRNA codon to figure out the amino acid. So CUA, right? CUA, leucine, LEU. Where is that? Here. Okay, so that's how to fill out the chart, and that's how to deal with tRNA. I think that you should be now able to complete the rest of the worksheet. In class, I mentioned to people that if they didn't want to do the mutations part, that's okay, because we are going to have a separate lesson on mutations. So if you are going to do this part, you will probably need to reference page 198 in the book so that you can see what a nonsense, missense, and frame shift mutation are. Have a wonderful rest of